So, Malati, there we go. Thank you, Malati. So I'm going to introduce today's speaker, Prof Gary Osthoff from University of Free State. Um, so there was a slight mistake in the email, but in the poster that was sent out, there's some correct information there. So just going off of that, Prof Osthoff was born in Germany and he grew up in Parys in the Free State. He studied at Northwest University and at the University of Free State. So his career started at the CSIR in Pretoria, where he researched snake venom proteins for 10 years. So that's also something we can be of interest maybe later on to talk about. He also completed a postdoctoral in the USA on enzymes, and he has been at the University of the Free State for 30 years, where his research has focused on food chemistry and biochemistry, and he has specialized in dairy chemistry. And he has over 85 publications and 5 book chapters. And today he is going to tell us all about African elephant milk. So I shall hand it over to you, Prof Osthoff. Thank you.
Thank you, Shane. Am I audible?
Yes, yes, you are.
Okay. Well, thank you, Shane and Metabolomics South Africa for inviting me to present this lecture. As you've heard, I was trained as a biochemist and I lived it out in the chemistry and biochemistry of foods, specifically of milk and cereal seeds. Food chemistry opened a whole new world for me to explore and apply biochemistry. Oh, what? Here we are. I will inform you about my research on the metabolomics of milk, specifically of African elephant. Since most of the audience is not acquainted with milk chemistry, I will have to introduce you to milk in general and elephant milk in specific to show why it became necessary to investigate the metabolome in order to find explanations for certain aspects of milk composition. We all know milk, and I'm sure everybody in the audience consumes it, maybe daily, or else some other dairy products will be consumed. The best known to us is cow's milk, but there are other domesticated mammals that serve as sources. It is fairly easy to obtain milk from domesticated animals and by the buckets full. Obtaining milk from wild animals, however, is much more difficult, and the amounts to be obtained are small. It is so precious to me that I refer to it as my white gold, and I try to squeeze out as much information as possible. Of the large mammalian phylum, information on milk has only been obtained from a few species. In this illustration, the degree of clarity of a picture presents the amount of data available of specific mammals. Boxed in blue are the species for which I had the privilege to contribute. Mammals fall into 3 groups: the monotremes, or the egg-laying mammals, the marsupials, or the pouched mammals. Those 2 we find in Australasia. And then we have the large group of the placentals. The placentals are the largest group with the most species. In times gone by, there were more monotremes and marsupials, but they are all extinct now. A subgroup of the placentals is the Atlantogenata, which consist of elephant, hyrax, dugong, and aardvark of Africa and Asia, and armadillo, anteater, and sloth of South America. The Atlantogenata form an early branch of the placentals, and less species of this group have survived to the current times. Hence, it seems strange to believe that the elephant and the hyrax are closest relatives. Regarding the milk composition, there are a few aspects that might suggest that the Atlantogenata differ from the Boreoerytheria. However, this evidence is still premature. What is clear is that the milk of elephant is very different. But further down the tree are also mammals with unique milk properties, the best example being the seal. Please remember this seal, because we will learn more about it later. Milk contains more nutrients than could practically dissolve in a volume of water. That is because it is a solution as well as a suspension. The major nutrients are divided into the carbohydrates, specifically lactose, and minor molecules such as glucose and oligosaccharides. The proteins consist of caseins, from which cheese is made, whey proteins, and a lot of enzymes. The lipid component consists of fat and phospholipids, and dissolved in the water is and the suspended particles is minerals and vitamins. The milk composition differs between species. To be noted in this table is the difference in composition of fat, the proteins, and lactose. Rhinoceros, as well as the whole Perissodactyla family, so far studied, have high lactose content. I want to draw your attention to the oligosaccharide content. Human mother's milk contains about 1.2% of oligosaccharides, while cow's milk contains 1/10 thereof, as do most of the other species. Elephant milk, however, contains even more than mother's milk, and a great part of the current presentation will elaborate on this phenomenon. The interspecies difference of milk composition contributes to the belief that the infants of each species have their own nutritional needs. The question is whether this is so, or whether there's a deeper reason for the difference embedded on the metabolomic or genetic level. When viewed under a microscope, milk displays its content of particles that are in suspension. The largest are the fat globules, which consist of an oil droplet coated by a membrane. The smallest particles are the casein micelles. Let us look at these micelles in more detail. The casein micelles are compiled by 4 different casein proteins: alpha S1, alpha S2, beta, and kappa. All 4 are needed in a specific ratio in order to form a micelle that will stay in suspension and will not precipitate. The kappa casein is protruding a negatively charged strand from the micelle, from the surface, and these repelling charges prevent aggregation. If these are removed by rennet in cheese making process, then the casein micelles will aggregate and cheese is formed. The structure of caseins and the micelle are not known. That is because they have a random structure and therefore cannot be crystallized for studies by X-ray crystallography. Some models have been proposed, like this one, and the sole purpose of these proteins is to serve as nutrients for the infants. The fat globule consists of a droplet of fat or oil that is synthesized at the Golgi apparatus and is covered with 1 layer of phospholipids. During pinocytosis, a double layer of from the secretory cell membrane is added. The globule therefore contains 3 layers of phospholipids together with membrane proteins and enzymes that come along with it. This forms a very large field of research of its own. The major sugar is lactose. Until 40 years ago, it was believed to be the only carbohydrate in milk. Improved analytical procedures led to the identification of oligosaccharides that are lactose molecules that have been elongated. The structures may be linear or branched, and the variety of composition of monosaccharides and the length and the branching types resulted in the identification of more than 250 types, most of which are species specific. Let us return to the taxonomic tree. The monotreme milk contains basically 0 amounts of lactose, with a large amount of oligosaccharides. Marsupial milks also contain large amounts of oligosaccharides and a low amount of lactose. The placental mammals, on the other hand, contain large amounts of lactose and low amounts of oligosaccharides. The odd one out is the elephant milk, which contains lactose as well as oligosaccharides in high amounts. The high lactose content is linked to the whey protein alpha-lactalbumin. We will return to this later. The reason for the presence of oligosaccharides in milk was provided by studying the monotremes and the marsupials. And it was found that the oligosaccharides do not serve the purpose of nutrition, but serve a protective function. The oligosaccharides serve as antigens that mimic the cell surface antigens of mammalian intestine. They bind to foreign microorganisms so that they can then be expelled. They also serve as nutrients for the gut bacteria, such as the bifidobacteria and lactobacilli. Due to this role, the oligosaccharides are also referred to as prebiotics and are one of the reasons why breastfed babies are healthier than bottle-fed babies. Milk metabolomics is a very new field of study. Because the study of milk metabolomics is still in its infancy, it is necessary to approach it through a non-targeted analysis. There is very little information available and comparison between literature is very difficult. And that is because the methodology of extraction, separation, detection, and identifications differ. Nevertheless, what is obvious is that most milks contain metabolites that are linked to cellular processes. There are also species specificities regarding the presence and absence of some metabolites and the amounts of others. And there are normally a large number of unidentified molecules. To demonstrate this, I present an interspecies comparison of some of my latest data. All samples were extracted and analyzed simultaneously, and as well as by the same methods involving nuclear magnetic resonance, NMR. Certain metabolites are absent in some species, others occur at high or low amounts, and there is a large number of unidentified metabolites. As shown in and this is also shown here by a 3D PCA from MetaboAnalyst. As was shown in a previous slide, the content of oligosaccharides is low, and the lactose content of rhinoceros is very high. It's double that of other species. You might have noted that I've made use of the link of other species in the past few slides. Many phenomena of milk could only be explained once comparisons between species were provided, one of which is the role of the prebiotic oligosaccharides. It is therefore clear that it is important that the milk of as many species as possible should be investigated, and not only that of human and domesticated animals, to eventually understand the nutritional role of all components present. This information eventually also finds application in the food processing industry. There are still many things to be discovered. I've investigated the milk of some 27 of the South African wild mammals, and the African elephant milk has revealed several very strange aspects of milk. I will briefly describe the main finding of the three major nutrients of elephant milk, and then zoom in on the aspects for which metabolomics were consulted to provide explanations. If you think the elephant is a strange-looking animal, wait until you have learned more about its milk. The first strange aspect of elephant milk is that its nutrient compositions keep changing over lactation. The fat and the protein content increases while the carbohydrate content decreases. This results in a constant increase in energy provision of the milk. Practically, it means that an older calf will be undernourished when fed by a cow that has a young calf. And while the interspecies comparison of milk metabolomes is still in our minds, let us look at the milk metabolome of the African elephant. This was also non-targeted analysis. It also contains metabolites of cellular processes, but some are at very high amounts compared to other species. It contains a large contingent of saccharides, some at very high amounts. Let us see whether we can link this unique metabolome to the other components of African elephant milk. First to be discussed is the proteins. The protein content of elephant milk we've seen increases over lactation. Casein is the major fraction, and it increases more than the whey proteins. The proteomics of the African elephant caseins is very interesting because it contains only beta and kappa caseins compared to other species. The reason is that the genes for alpha S1 and alpha S2 caseins have been lost during evolution. I do not want to elaborate on that because the interconnection of these proteins and the metabolites is limited. However, when metabolomics of the African elephant milk was investigated, some interesting answers were obtained. The presence of succinate was no stranger in the milk, as it was also reported for other species. This is an indication of metabolic activity of milk gland cells, which involves the citric acid or PrEP cycle. And in this case, the increase of succinate content over lactation indicates that the metabolism is stepped up, specifically providing energy and metabolites for the macronutrients, such as amino acids for the synthesis of milk proteins. The next is the fat composition or fat content of African elephant milk. Fatty acid composition of the triglycerides of milk fat may vary in length and saturation. These have three origins: the diet of the mother animal, saturation and desaturation in the mother animal's body, and then there's de novo synthesis in the milk gland. In ruminants only, contain short chain fatty acids, butyric and hexanoic acids. And these are produced by the rumen bacteria and therefore a part of the dietary obtained component. The medium and long chain fatty acids may differ due to the diet, with some species specificity observed. The whole Alcelaphine family, which contains the blue wildebeest, black wildebeest, blesbok, and all the types of hartebeest, display a high content of medium length fatty acids. And the primates and carnivores have a high content of long chain polyunsaturated fatty acids. Elephant milk fat contains a high content of octanoic, capric, and dodecanoic acids, irrespective of external factors such as the diet and season. This interspecies differences, again, suggests a deeper reason at metabolomic or genetic level rather than the superficial reasoning of fulfilling the needs of the infant. We've seen earlier that the milk fat content of the elephant changes over lactation. The fatty acid content also changes. This is quite unique in the mammalian kingdom, specifically towards a high content of capric acid at the cost of long chain fatty acids. One answer to the species specificity of chain length of milk fatty acid may lie in the properties of the enzyme in the milk gland involved in the synthesis of the fatty acids. In liver and adipose tissue, fatty acids are normally elongated, normally not elongated by the fatty, where am I now? Sorry. In the liver and adipose tissue, fatty acids are normally not elongated by the fatty acid synthase beyond the length of 16 carbons. In the milk gland cells of some species occurs another enzyme involved in fatty acid synthesis. It is a thioesterase that may interrupt the elongation process by jumping in and terminating the fatty acid length at 8 to 14 carbons. The specific length preferred by this enzyme may depend on the species. This has been studied in rabbit and goat. So the role of this enzyme in fatty acid length has not yet been investigated in the elephant, so that this explanation is still speculative, but based on rabbit and goat milk, a probability. In elephant milk, this results in triglycerides with a composition of a limited number of fatty acids, as was determined by chromatography and mass spectrometry. Instead of having over 100 possibilities of fatty acid combinations, such as in the cow's milk, the elephant milk contains only 12 molecule species of triglycerides, mainly 4 types. And this also, limited number of triglyceride species allows for a very dense packing of fat crystals during cooling. The medium chain fatty acids result in a fat that has a low melting point and a narrow melting range. Butterfat from cow, as we all know, is solid at room temperature and is a soft solid at temperatures as high as 30 centigrade. The elephant milk fat forms a hard crystal fat on crystallization, but it melts at 22 centigrade. This was determined by differential scanning colorimetry and X-ray refractometry. Right, regarding the milk metabolites, there's a more pronounced interconnection with the lipids than for the proteins. The presence of succinate, as we've seen, and its increase over lactation indicates the stepped up metabolism of the milk gland cells, also serves to provide de novo synthesis of fatty acids for the milk fat. The capric acid content in elephant milk was high, much higher than observed in goat's milk, which also contains a high amount of other mammals than other mammals. This indicated demand for this acid in the milk fat synthesis, as we've seen in a slide or two ago. Lastly, the content of choline and phosphocholine was high in elephant milk, indicating the need to synthesize membrane material for the fat globule. The choline content was also found to increase over lactation, indicating the need to provide sufficient membrane material to accommodate the increasing amounts of milk fat. The last macronutrient of elephant milk to be discussed is the carbohydrates. We've mentioned earlier that the presence of alpha-lactalbumin, the presence of lactose in placental mammals' milk has to do with the whey protein alpha-lactalbumin. The monotremes and marsupials do not have that protein because it had not developed in them during evolution. The seal also does not contain lactose due to the absence of alpha-lactalbumin, but this animal has lost the gene for this protein. The contrary is observed in its closely related cousin, the sea lion. Alpha-lactalbumin evolved from lysozyme, which is one of the ancient proteins that is found right through the animal kingdom. Alpha-lactalbumin is part of the lactose synthase complex. It consists of 2 proteins. It helps alpha-galactosyl transferase to speed up lactose synthesis and also to transfer the lactose across the cell membrane of milk gland cells. Other transferases will use lactose to add additional glucose, galactose, and fructose molecules to form oligosaccharides. Due to the absence of alpha-lactalbumin in the monotremes and marsupials, lactose is synthesized slowly and is also transported slowly. There is thus ample time for the transferases to elongate them into oligosaccharides. The question is, why are lactose as well as oligosaccharides present in high quantities in elephant milk? Is it due to a malfunctioning lactose synthase activity or to a highly active transferases? So by deriving amino acid sequence from the gene sequence, it was able to do some protein modeling. It was found that the African elephant alpha-lactalbumin is most probably folded into the same structure as that of the same protein from 4 species, of which the structure was described by X-ray spectroscopy. The same was concluded for the alpha-galactosyl transferase structure, and then the lactose synthase complex when those two proteins were combined. All the necessary amino acids side chains involved in van der Waals interactions were in place, as are the ones in the active site. This means that the lactose synthesis by African elephant milk gland cells should therefore progress with the same kinetic properties and as in other placental mammals. We've seen earlier on that the total saccharide content of elephant milk is decreasing over lactation. In more detail, lactose content is decreasing to very low levels, and the amounts of oligosaccharides then become the major saccharide fraction. This then also shows a shift from lactose as energy source to fat and protein, as we've seen previously. The decrease in lactose over lactation we've also quantified, quantitatively confirmed by the metabolomics technique, and it followed exactly the same concentration line. The oligosaccharide fraction consists of a collection of different structures. 16 of these structures have been described in elephant milk. Some of these are unique to elephant. At the time when this research was carried out, it was not possible to delve deeper into the oligosaccharide properties. They were, for instance, not quantified. Peak size, as obtained by light absorbance, was not an estimate of quantity due to the different molecular lengths, and quantification had to be carried out by chemical derivatization and quantification of the marker. With the metabolomics method by NMR, it became possible to simultaneously identify and quantify molecules. That is, if the NMR spectra are available as reference in the data bank. With this method, it was possible to identify and quantify 5 known and 4 unknown oligosaccharides in the African elephant milk. The ones identified were in the data bank because they occur in most other milks as well. Unfortunately, it was not possible for us to retrieve the spectra of the previous work from electronic stores for our desired analysis. It is therefore possible that this DF-para-LN non-O's, which is unique to elephant milk, was one of the unknown oligosaccharide detected by the NMR analysis. Nevertheless, the metabolomics study by NMR revealed some interesting facts. As per the previous study, both lactose and oligosaccharide content decrease. The decrease of the oligosaccharides seemed to drop lower and much steeper than was observed with the chromatographic analysis data, which I've shown a few slides ago. The reason might be because this metabolome graph was compiled by mathematical addition of the amounts of the identifiable molecules only. The unknowns were not included because they could not be quantified according to a reference. The other important result obtained is that there's a dynamic change of the content of individual oligosaccharides over lactation. Some decrease, others increase, and a third group may stay constant. This accounts for both the identified as well as the unidentified saccharides. It is of specific importance to have observed that some of the oligosaccharide contents increase over lactation, as they may play a role in the protective function in the infant elephant's intestine and the settling of the CSIR bacteria. A colleague of mine is currently writing up research on the microbiome of the African elephant intestine, which supports this. The increasing of content of succinate serves as proof of increased metabolic activity of the milk gland cells to produce the nutrients, proteins, and fat. Although its increasing tendency does not support the decrease of the saccharides, the increased metabolic activity is also required for saccharide synthesis. The glutamate content of the milk was also found to be exceptionally high when compared to other species. Glutamate is a glucogenic amino acid that is metabolized to alpha-ketoglutarate. This enters the citric acid cycle, is then changed to oxaloacetate, and which is then utilized for gluconeogenesis. The high glutamate content is therefore supportive of the high saccharide content of elephant milk. When the biosynthesis steps of oligosaccharides are added to the dynamic changes of individual saccharides, it is possible to derive the changes in activity of individual transferases. In the example shown here, it can be derived that most of the 3-fucosyl lactose is converted to lacto-N-difucohexose 1 during early lactation, but less so during late lactation. One can only wonder what role this may play in the settling of the intestinal bacteria in the elephant calf during the different lactation stages. Furthermore, the transferases involved in the polymerization may now also be investigated. According to all the possible glycosidic bonds observed in milk oligosaccharides of elephant milk, there may be at least 8 transferases involved. So far, no detail is known on this, and the metabolomics technique could be of use to solve the riddle of the simultaneous occurrence of high contents of lactose and oligosaccharides. Those of you who are involved in clinical metabolomics are acquainted with the combining of metabolomic data of blood serum and urine for interpretation of health conditions. Urine is a sink for certain metabolites. Because a great deal of the milk nutrients depend on molecules transported in by the blood, we thought it good to compare elephant blood serum metabolites to find out whether there are metabolites that are transferred to and fro between the milk gland cells and blood. This was a targeted analysis, and all the answers that we would have liked can therefore not be obtained for the to and fro movement of molecules. However, the serum metabolome of our group of African elephants could be divided in 3: males, females, and lactating females. The blood serum showed no great difference between the metabolomes of males and females. However, the lactating females displayed a high content of 2-hydroxybutyrate and L-lactic acid, and a low content of D-glucose, which indicates a higher metabolic activity for the energy demand of milk production. The variations of pyruvic acid, citric acid, and succinic acid indicated an increase in the expression of glycolysis enzymes with a simultaneous downregulation in the Krebs cycle and significant induction of lactate dehydrogenase as a means to increase lactate. To conclude, we've seen that the milk composition of African elephant is unique. The information on this milk challenges the current knowledge of lactation biochemistry, and it seems as if the African elephant milk may serve as a model of comparative studies of certain aspects. An interconnectedness can be observed between milk components, such as the fat and carbohydrates, and several metabolites, and explain interspecies differences. The metabolomics method methodology can contribute to the study of the dynamic biochemistry of carbohydrates of milk, specifically regarding the synthesis of the prebiotic oligosaccharides. The information I've presented here was generated over 20 years. If science finds methods of how to extend the life of humans, let me know. I would like to spend some of this added time on unraveling a couple of more phenomena of milk. Thank you for your attention.
Thank you so much, Prof Osthoff. It is incredible how much information you've obtained from a very complex creature. And yeah, I think you're like the only person in the world who has this knowledge. And we are very privileged to share it from you. So thank you very much.
Thanks for the compliment.
Um, what I always encourage our speakers just to, in the chat box or in the presentation, just to share an email that they can communicate with you if they have any ideas to contribute towards your understanding or want to collaborate with you or anything like that. So I think if you do not have a final slide with your email address, maybe you can put it in the chat box so that
Okay.
people may communicate with you. And to the audience, if anyone has any questions for our speaker, you are welcome to either type in the chat box or you can raise your hand and ask directly. So we do encourage communication with the presenter. He is obviously a very intimidating man of knowledge in this field, and we are very little knowledge ourselves, but it is very interesting the way you describe it. And I have had the privilege to work with you, Prof Osthoff. It has been very interesting looking at elephant milk. And a lot of what you've shown is in millimolar concentration, which are it's very high concentrations.
Yes,
So
some are indeed high concentrations.
so just make note of that, everyone. It's not micromolar concentrations, are millimolar. These are very high concentrations. So it's a very interesting spectra that we obtained for the NMR. And I believe, Prof. Osthoff, you will be retiring in the near future. Would anyone be following in your footsteps in your research? I hope it does not go with you.
Unfortunately, there is no one who's following.
I'm hoping someone in the audience, oh, we do upload these onto YouTube, so maybe someone listening on YouTube would get in contact with you and want to continue your research, because 20 years of research, you're coming to some interesting theories and you definitely wanted to test that further. So this is something we should continue research into. So anyone listening in the YouTube land, um, perhaps you can contact Prof. Osthoff and see how you can continue his research. And I see you've had the wonderful opportunity to interact with these animals as well. So you're quite blessed in that aspect.
Thank you.
So anyone in the audience, do you have any questions? Please. Kim, your hand is up. Please ask our presenter.
Thank you very much, Professor Osthoff, for a very wonderful presentation. It was so illuminating, and your work sounds so exciting. I think I'm going to give up my job and come and follow in your footsteps instead. I'd just like to ask you, how are you actually procuring the breast milk, number one? And secondly, I noted that some elephants were on contraceptives. What's that about, and where are these elephants? Thank you.
Thank you for your compliments, also for the question. The collection of milk from wild animals is by chance. With ethics, as we all know, the frustrations of ethics, it will not be possible to specifically dart animals to obtain milk. So what I've done is I had contact with other researchers and veterinarians that when they were darting animals, milk could be collected. And yeah, even from dead animals, I've been out with harvest hunters, and luckily these guys don't mess. They shoot for headshots, and within 20 minutes after death, then milk can also be collected before it changes biochemically. Now, elephant, it is not as easy to milk an elephant, not that it might kick or anything. It's one has to fool the elephant cow. She doesn't just give you milk like a cow does. The milk letting has to be stimulated by suckling. So what the handlers do is they sneak in when the calf is drinking on one teat and then steal milk from the other. But interesting. And yeah, most of the elephants in captive or small parks are on contraceptives lately. Elephants breed very easily, but of course they eat quite a stack of food, and it is expensive to keep them. So the small herds on small properties or premises are then, well, they like to keep the numbers in control.
Thank you very much. Very fascinating topic. Thanks so much.
Are there any other questions from the audience? So I think there are lots of unanswered questions, lots of dark matter concepts that we still haven't investigated with elephants, and how we can approach it, and the health benefits for us as humans as well. So much to think about.
If I, just for interest's sake, okay, it is blocked now. The whole idea of working on milk metabolomics was initiated when I had a discussion with Prof Mini, who is now retired. And that dealt specifically with the fatty acid length of the milks. And he said this would be very good news if we could have milk with short chain fatty acids or medium chain fatty acids, such as we find in the L-cellophines, the Blesbok and so on, or even better, the elephant. Because you have this one metabolic disease, I forgot the name now, where the people cannot digest long chain fatty acids. However, they can digest short chain fatty acids. Some cannot digest fatty acids at all, some of those patients. And he mentioned that that would be a good idea to delve into this. So if one can explain why certain mammals are rather synthesizing shorter fatty acids than the longer ones, maybe it is that thioesterase that is involved. But at least if one can understand that mechanism, one could change genetically or by enzyme induction or whatever, could change cows to produce milk with shorter fatty acids. At least one can help some patients, some infants that have that digestion disorder.
Yes, there are lots of theories you are throwing out there that people need to test.
Yes. So the work as I'm approaching it is a bit different from the clinical studies, where there's already a data bank of a baseline metabolome available. I have to create the baselines and then start comparing. And as we've mentioned, that the methods and analyses and everything has to be 100% equal. Otherwise, comparisons would not be possible.
Mm.
If someone from the audience could contribute to this, I would appreciate it.
I hope so. I'll even from viewing the YouTube video later, anyone who has any ideas to think about, I encourage contacting Prof Osthoff about it. I'm sure he will be happy to talk about it. So there's a question in the chat box. How do you get around the ethics when publishing?
Yeah, well, the normal way, you have to apply to ethics committee and to the Section 20 research. All that paperwork has to be in line. Mine has lapsed now. I'm only allowed to work on the samples that I've collected before a year ago, because there were some changes in the ethics rules. But I'm also connected with other research groups where work on other animals such as rhinoceros and giraffe are linked. So we've added my research there. So yeah, that's how we try to get across the problem.
Okay, so if there aren't any other questions, I'm going to thank our speaker, Prof Osthoff. You are one of a kind researcher, and we are very fortunate to have you present. Thank you very much. We appreciate it.
Thank you, Shane, and thank you to the audience for listening.
Okay, all the best with the rest of your research, Prof Osthoff. I hope you do get some communications from this.
Yes, thank you.
And you can then test your theories going forward.
Okay, bye everyone. Enjoy